All right, welcome back. Part 2 of Heredity 5. All right, so we did a little bit about cystic fibrosis. And now let's take a look at Tay-Sachs. Tay-Sachs is a really rare autosomal recessive genetic disorder. So autosomal means that it is on the autosomal chromosomes, not the sex chromosomes. And we're going to talk about things on the sex chromosomes in the next screencast in part 3. So Tay-Sachs is on autosomal. It causes deterioration of nerve cells and then loss of mental and physical abilities. This starts to happen about 6 months of age and usually results in death by the age of 4. So this is what happens on the cellular level for Tay-Sachs. Normal lysosomes are able to digest this biochemical called ganglioside GM2. So they digest that, and then the digested portions in this residual vacuole fuse with the cell membrane, and those waste products are excreted out of the cells. And this will happen in brain cells, nerve cells in the brain. For Tay-Sachs lysosomes, the digestive enzymes, they have all of them except one that specifically digests ganglioside GM2. That one's called hexoaminidase A. You won't need to know the names of these, but just in general, if you're curious, this is what happens with Tay-Sachs. So ganglioside GM2 gets put in a digestive vacuole. Because the ganglioside GM2 is not digested, then these residual vacuoles accumulate. So the cell starts to fill up with all of these residual vacuoles, and that causes damage to the cells, so you don't have any exocytosis of those waste products like you do in a normal cell. Okay, now this is an interesting one because in the general United States population, it's about 1 in 320,000 newborns have Tay-Sachs. And what they do is they will genetically test almost every newborn right away for certain disorders, and Tay-Sachs is one of those. So when a newborn gets a blood sample and then they go and they test that, they're looking at, they have tests that will detect certain genetic disorders, and Tay-Sachs is one of those. The interesting thing about Tay-Sachs is the incidence in certain groups, what we would call cultural groups, or groups of certain lineage or heritage, have a higher incidence of Tay-Sachs than the normal population does. So if you look at Ashkenazi Jews and French Canadians and Cajuns, their disease incidence is much higher, about 1 in every 3,500 newborns. In fact, 1 in about 27 or 1 in about 30 Ashkenazi Jews carry the disorder, so they're heterozygous for Tay-Sachs. Irish-born Americans, or even people of Irish descent, it's usually about 1 in 50 is a carrier for Tay-Sachs. And then French Canadians and people of Cajun descent would have a similar incidence, where about 1 in 30 are carriers for Tay-Sachs disease. So this was an interesting one that shows that some groups are more prevalent in certain genetic disorders. And that would relate to what we talked about earlier with consanguineous marriages. If people of these groups only married together, then it would increase the chance of having these recessive allele disorders show up in their offspring. Okay, let's try sickle cell disease, I think is next. Right. Okay, so sickle cell disease is a more common one than cystic fibrosis or Tay-Sachs. It affects about 1 out of 400 African Americans. So this is another one that is more prevalent in a certain group. The incidence in all Americans is lower. I don't know the exact number, but I know that the incidence is much lower. This disease is caused by a mutation called a substitution. And this substitution occurs in one single amino acid sequence in the protein hemoglobin, in the code that makes the protein hemoglobin for red blood cells. So people who have sickle cell disease have physical weakness, pain, organ damage, and even paralysis can occur. So this is what will happen with sickle cell disease. Here I have a picture of some red blood cells in capillaries, and these are the normal red blood cells, these nice round donut-shaped red blood cells. These are these are their structure fits their function. They're able to slip through. Normal red blood cells are able to slip through these capillary tubes or these capillary vessels one at a time rather nicely, and they don't. Their shape allows them to just go through really slick-like. Sickle cells, they're misshapen, so they have this kind of crescent shape, so that's why they're called sickle cells. So what they can do is in blood vessels, they can create blockages so that the blood doesn't get through. They can also damage tissues. You can get coagulations that occur and bursting of normal red blood cells. So sickle cells can do a lot of damage to tissues. Now, one thing that we'll talk about when we get into evolution is a heterozygous condition for sickle cell anemia, where people have some sickle cells, misshapen sickle cells, and some normal cells. And they're able to survive bouts of sickness of malaria. And we'll learn a little bit more about that later, so that's kind of a clue for something that we'll learn about later this year, how sickle cell disease was more prevalent in areas where malaria was prevalent, because it was a pressure that selected for that sickle cell disease because it added to resistance to malaria and therefore showed up more commonly in the population. But we'll put that on pause for now, and we'll come back to that later when we get into evolution. All right. Now, not all disorders are recessive allele disorders. Some are inherited dominantly. Okay, we call those dominant disorders. They're caused by dominant alleles, and those that lead to a lethal disease are rare and usually arise by a mutation. Imagine that if you had a dominant allele disorder, you'd be dead before you could reproduce, and so you wouldn't be able to really pass that disorder on. There are a couple of ones that are non-lethal. So let's talk about the first one in your reading guide. With dominant genetic disorders, the important thing to remember is that it only takes one mutant allele for somebody to have that disorder. So if they're heterozygous or if they're homozygous dominant, then they are going to have that disorder. And sometimes you can tell by looking at pedigree charts whether those disorders are dominant or recessive allele disorders. So let's take a look at the first one that your book talks about. This one is called achondroplasia. It's a form of dwarfism. So notice that the heterozygous parent right here has that disorder because this parent has, and it looks like he's the father, he has the big D allele, or the dominant version of that gene. Okay, the mother is recessive for both, so she's normal. And this goes into what we talked about a couple of times ago, where the dominant version of the gene does not necessarily mean it's the most common version of the gene. I know we talked about peas, and we said, I was thinking about this today. Peas were on the menu for lunch. We were eating roast beef with peas, and all my peas were green, and I remembered that the reason why that most peas are green is because the dominant version of the gene is not necessarily the most common version of the gene. Sometimes the recessive version of the gene is the one that's most common in the population. So that's why most peas are green, is because the recessive version of the gene is the one that's the most prevalent. It's the one that gets passed on the most instead of the dominant version of the gene. So some, but few peas are yellow, and most peas are green just because there's a lot more little Y's out there in pea plants than there are big Y's. Okay, the same thing works for these dominant allele disorders. There are more little letters present in gene pools than there are big letters. Okay, so this is how that would work. This father can either donate a big D or a little d to his children. The mother, her eggs can only be little d since she is normal. So children from this mating right here, or this marriage right here, have a 50-50 chance of being dwarfed compared to normal. Okay, Huntington's disease. This is a nervous system degenerative disease. This one is really tricky because it doesn't have any physical outward effects until the individual reaches about 35 to 40 years old. So by the time somebody knows that they have Huntington's disease, they usually already have children, and they may have passed that disease on. If they're heterozygous, if they would be big H, little h, then there's a 50-50 chance that their kids will inherit that disease, assuming that the other parent is normal. And if they are homozygous dominant for Huntington's disease, then their children have a 100% chance of inheriting the disease, even if they have a normal mother, because they will be heterozygous for that trait, since it's a dominant allele disorder. Okay, let us skip to genetic testing and genetic counseling. So, sometimes if there is a prevalence of a genetic disorder in a certain family, people will look and use genetic counselors to find information about whether they have a chance of passing on this disorder to their children. And they can make an informed decision about whether to have children or not based on the chance that their child has of inheriting this disorder. So people who test and help people decide on these are called genetic counselors, and they do genetic testing. So they would take blood samples from people, and then they would look at their chromosomes, and sometimes those chromosomes can give them clues, and they can test the genes in those chromosomes that will give them clues as to whether they have or if they are a carrier of a certain genetic disorder. So if you're thinking like Tay-Sachs, if you were one of the people of that specific Jewish descent, and you were wanting to marry another person of that Jewish descent, you could be tested for those genetic disorders by a genetic counselor, and then the genetic counselor could tell you what the chances were that you would have a child with Tay-Sachs, so you could make an informed decision about having children. Okay, so let's take a look at the different tests that are available to identify carriers. Actually, here's a test that's done after fertilization has occurred, so testing of a fetus. There's two ways to do that. One is called amniocentesis. So in amniocentesis, the fluid, the amniotic fluid that surrounds the baby, is taken. So a needle is injected into the uterus, and a sample of fluid is taken. This one can be risky because it can damage the amniotic sac, and it could possibly cause the fetus to abort. So typically this is not done unless they really want to make sure that this fetus does not have this disorder that may harm the mother. And they centrifuge the amniotic fluid, and they take the fetal cells that are floating around in this amniotic fluid, and they grow it out, and they do a karyotype of that to see if they have certain disorders that can be detected by just doing a karyotype. and sometimes they can be detected by